Hi, my name is Tin, and I worked in the Nelson Lab, and in the Nelson Lab we focus on plant stress. And why it's important to understand plant stress is because there is constant change in the environment that is constantly happening, and we want to understand how to help plants withstand those types of stress. And so my topic is high throughput phenotyping of Arabidopsis mutants under stress. And so in the lab, I had two projects. I had one that focused on oxidative stress, and then I had one that focused on drought stress. In my first project, we received seeds from that were Arabidopsis, and it had tDNA insertions. We mainly focused on link RNA, but they were associated with oxidative stress. So one aspect of my project was root growth, and then another aspect was germination assays. And so in the root growth, we plated the seeds on plates that had hydrogen peroxide, and the plates had different amounts. Some had 5 millimolars, some had 2, and some had 0. And so from the daily imagings we took and the root growth analyzing we did on image J, we noticed that as we increased the amount of hydrogen peroxide, the root growth decreased. And that was the same case for the germination assays. So as we increased the amount of hydrogen peroxide on the circular plates, the seeds that germinated decreased. And so we noticed that the mutants did better than the wild type, and that's the same case for the germination. Some mutants did better than a wild type. And so what we concluded from the first project is that we should further investigate the mutants and make sure the reason why they did better was because of the gene and not something else that happened in the experiment. And so moving on to my second project, which is drought stress. We used a camera that took an image of the Arabidopsis plants every 30 minutes for 2 weeks straight. And we used a computation tool called PlantCV. And so what we did was we programmed the computation tool, PlantCV, to only look at the rosette region of the plant. And so from all those daily imagings that we got, we ended up with a result. So we had two types of environment. We had a controlled environment, and we had a drought environment. And if you zoom up on the graph, you notice that the red dotted line is the mutant, and the gray dotted line is the wild type. And so our result was in the controlled environment, the wild type did better, but in the drought environment, the mutant did significantly better. So that is something important for us to look into because we want to understand why that mutant had such a good drought tolerance, because it would be useful for other plants. And so that was my project, and my project is like the first step, and there will be further steps to investigate into the mutants that did well. I guess the funnest thing for the Nelson Lab is we had lab meetings on Wednesdays, and sometimes we'd go on coffee runs, and sometimes we would get pastries from Gimme Coffee, and it was really good. And everyone in the Nelson Lab was really nice, and some of the other mentors that weren't even my mentor, they gave me some goodbye gifts, and they were really sweet.